Hey everyone, and welcome back to 45-7. I know it's been really a long time since the introductory video, but we're back and we're getting straight to work, I promise. So, here's the plan. I've decided not to teach the lessons from scratch like a boring bio lecture. Instead, we're going to think of this as a revision session with your study partner. You're just making sure you got every point 100%. Yes, we're going to treat this like we're preparing for the exam right now, since May 2026 exams are just coming up. And I have picked this topic specifically because it is a hands down one of the most challenging topics in the syllabus. If you've looked at past papers, especially paper 1A, you know that the IB loves to ask tricky questions about enzymes, directionality, and specific steps. And since this is the building block of the protein synthesis, you must and should understand it step by step, okay? So we're going to use the IB Biology Guide as our reference and checklist to make sure we don't miss a single mark. So first, let's ground ourselves. We're here now in Theme D, Continuity and Change, specifically D1.1 DNA Replication. And this whole theme, alright, talks about how life maintains stability while allowing us for evolution. DNA replication is the most ultimate example of continuity, however, keeping our genetic code safe from one generation to the next. Make sure that in each video we're going to have the guiding questions which are provided in the IB guide. You might ask me why they're just read only. No, they actually have a very large effect on your absorption to the upcoming ideas and information. So these are two questions: how is new DNA produced, and how has knowledge of DNA replication enabled applications in biotechnology? Alright, so if you want to test yourself that you have already known the lesson, which I really prefer because we're just revising, alright, so pause the video and answer them. So if you answered these, congratulations, I'm clapping for you, and if not, please don't worry, that's what we're going to do for today. So firstly, let's active recall some things from A1.2. We've already taken the structures, not in the channel, but you must have done this with your tutor at school, with your teachers. So what would you recall? You would recall the structure that we have already known that DNA is a polymer composed of monomers called nucleotides, alright? And that the nucleotide contains a 5-carbon sugar called deoxyribose and a phosphate group and a nitrogenous base, and that this is the basic structure for DNA. Here is the IB points that I have told you about. Okay, as you see, we're going to go through point by point so we don't miss any single thing provided by the IB. And at the end of the video, please, I want you to download it from the IB official site and make sure that you understand every single point, because it's really crucial that you go through it before the exam. Believe me, the IB never gets out of its guides. Believe me, I was an IB student. Firstly, and before starting our revision, I want you to watch this video. Either you're going to recap the information before we start or get a background of what we're going to talk about in today's lesson. Let's start.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called Helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called Primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA Polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Alright, let's check off the first point for today's video. So, what is DNA replication? Replica is simply an exact copy of something. So DNA replication is simply the production of exact copies of DNA with identical base sequences. But why do we need this? Firstly, for reproduction, because cell division can occur and pass our genetic information to our offsprings, but actually not just for this, but also for growth. You can't make new cells if you don't have the same instructions, and also for tissue replacements, right? When you scrap your knee, the new skin cells need the same DNA as the old ones to get you what you want. And the key takeaway here is that copies must be identical, because any mistake in such a process means mutation. So, for the next one, we have the conservative nature of DNA. Here, look at this diagram. When the DNA replicates, we don't just build a whole new double helix from scratch. No, we just split the old one. One strand is the parent strand and the other is a newly synthesized one. And this is huge for accuracy. The original strand just acts as a template for the new one, and this ensures that there is genetic accuracy. But what also helps in giving some stability is the complementary base pairing that occurs where adenine binds with thymine by 2 hydrogen bonds and guanine binds with cytosine using 3 hydrogen bonds, and this is the secret to the high degree of accuracy and stability. The DNA polymerase here does not actually ensure by guessing. No, it actually solves a puzzle. It gives the missing pieces to the right place, reducing errors. And why this matters? Because it maintains the integrity of our genetic information across generation and prevents any mutation that could harm cells or organisms. And here let's talk about our enzymes. For the standard level, you focus on two main players, the helicase and the DNA polymerase. Think of the helicase as a zipper or a DNA unwinder, alright? It breaks the hydrogen bonds between the complementary DNA bases and unwinds the double helix. And this creates the replication fork where strands can be newly synthesized as we see here. And the DNA polymerase is our builder. Okay, it adds new nucleotides. It creates the phosphodiester bond, linking nucleotides into a new strand. This gives the strong backbone of the bonds. Alright, and a crucial point, take care that the DNA polymerase always works from 5' to 3'. Okay, we'll talk more about that in the HL section, but for you standard levels, just remember this direction. Let's jump to a very important piece in our lesson today, and it is the PCR and Gel Electrophoresis. Now, you should know that the IB loves asking about how we can use this knowledge in the lab. And since biochemistry and biotechnology is now one of the most arising fields, you should care about them too. First up, the PCR, the polymerase chain reaction. Think of the PCR as a molecular photocopier. Yes, it really photocopies. If you have a tiny drop of blood at a crime scene, you use the PCR to amplify it, make millions of copies from it. It uses distinct steps, okay, and distinct temperatures. First, you should know that we use primers and the Taq polymerase. It is a very heat stable enzyme that reaches 95 and never denatures. Okay, and primers, they're just short DNA sequences. They bind the target regions on DNA and make it as if just labeling something to start from. So, the temperature changes stages. First step is denaturation, just separating the strands. Then annealing from 50 to 65 Celsius, where just primers bind, alright? And then the extension at 72 Celsius, where the Taq polymerase adds nucleotides to build the new strands. Okay, and this cycle would be repeated a lot to exponentially increase the DNA copies. Here is denaturation, then annealing, then extension. The gel electrophoresis, once we have the DNA, we separate it. But how do separate the DNA? By its size. Yes, DNA is negatively charged, and so it moves towards the positive electrode. Sorry, this is negative and this is the positive electrode. Alright, so small fragments move faster and even further. Alright, and the gel just stucks the large fragments and make them move slower, and this creates the board. Here it was. Wait, in the yes, this is the ports which the gel electrophoresis makes. Sorry, yeah, and this creates the barcode pattern you see in textbooks and in crime scenes. Just like this, they take pattern and make it compare, like the, what would I say, the parent and a child, or maybe a dead one in crime scenes and the one who killed him, the serial killer, and the one suspected. Okay, And this is a summary table between the PCR and gel electrophoresis. PCR amplifies specific DNA sequence. It makes exponential copies of it, and the gel electrophoresis separates them by size. And the key features that you should know that we use primers, stack polymerase, and some temperature cycles in the PCR. However, in the gel electrophoresis, DNA moves through gel by its size, whether it's small moving faster or large moving slower. So why do we use this? We do this for DNA profiling, whether it is for forensic investigation, like who committed the crime, who is the serial killer, or a paternity test, who is the father for the child. We're comparing these bands. And pro tip for exam reliability, how do we make sure we don't convict the wrong person? Like maybe it's just similar DNA, for example. That's why we use multiple makers, okay? Multiple DNA makers, and this reduces our chance of false matches, okay? The more makers you check, the lower chance of false match, okay? And this is really important for ensuring the trustworthy. And warning, here is the higher level content. If you're an SL student, you can chill for a second, or maybe if you're curious, you can stay and learn. But HL students, listen up. This is where paper 1 tries to trap you, and we need to get detailed. Right. Firstly, we're going to talk about directionality. We established that DNA strands are anti-parallel, right? One runs from the 5' to 3', and ones run from the 3 to the 5'. And the golden rule here is that the DNA polymerase can only add nucleotides to the 3 end of a growing strand. And this means synthesis should always happen from the 5' to the 3' direction. It can never work the other way, okay? Here we can see this is the 5th end of the growing strand from 5 to 3 end, and here is the DNA polymerase should be working here from the 3 strand of the template strand. So just be careful, because here comes a tricky question. He would tell you which side would the DNA polymerase work. You would say from the 5 to 3 direction, but if he asked you in which end does the DNA polymerase add the nucleotides, you would answer what? The 3' growing end. Take care for this point. Alright, now for the leading and lagging strand. Because of that 5 to 3 rule, as we have mentioned, okay, we have a problem on one strand, the lagging strand. The leading strand, this one is just very easy. It points towards the replication fork, with the replication fork, so it goes from 5' to 3'. It works continuously, and everything is very peaceful here. The problems comes on this one, the lagging strand, where it's just synthesized discontinuously because it points away from the replication fork. We have to build it in chunks, and these chunks is called the Okazaki fragments, one by one. Alright, this synthesis is called discontinuous. Okay, so the leading strand, the 3 end is always exposed so that the DNA polymerase 3 can work continuously without stoppage. But here, the 3 end is not constantly exposed. You're going away from the replication fork, so it works discontinuously, making Okazaki fragments. And now let's know some enzymes in the prokaryotic DNA replication. Just take care of such a word. Okay, for HL, it's not just helicase and polymerase anymore. You need to know the whole squad, okay? Specifically, as I said, in prokaryotics. Firstly, the DNA primase, okay? It lays down a short RNA primer. And why? Because DNA polymerase needs a starter block, a label, as if a sign to just work from here. Okay, it labels for the DNA polymerase to start from its place. Okay, and for DNA polymerase 3, it is one of the main enzymes. It's the main builder. It extends the strand by adding nucleotides. And remember in which direction? The 3' direction? No, from the 3' end, making it goes from the 5 and 2, sorry, it's not working, to the 3 end direction. Alright, and the polymerase 1 is the clean up. Let's write it. It's really the clean up crew. It removes the RNA primers, those labels and signs, and replaces it with DNA nucleotides. And then DNA ligase, our glue. Alright? It joins the Okazaki fragments together by forming phosphodiester bonds. And a note, I know it might not be mentioned in every single textbook, but it came in a post paper. The DNA Gyrase relieves the nicks or the strains ahead of the fork so the DNA doesn't snap. You would say, I don't know what you're talking about. Okay, let me clear it up. So here is the DNA. But I don't want you to think of it as a DNA. I want you to think of it as a braid. If you're a girl, you can imagine it. If you're a boy, try, okay? You're having a braid. And when you're unwinding your braid, knocks can happen here, and knocks can happen here, and helicase might stop unwinding it. Here is the helicase. And a nick happened, alright? So DNA Gyrase comes to uncoil this knots and free it up for the helicase to continue. Hope you got it. So here is a summary for all the enzymes. Okay, here is our DNA helicase unwinding our DNA double helix, and here is the DNA polymerase 3 adding nucleotides from the 5 direction to the 3 direction, but started in the 3 end. This is our leading strand going continuously without stoppage, and here is our lagging strand going discontinuous. Our Okazaki fragments are here. Our RNA primers labeling for the DNA polymerase 3 to start after 8, and here is the DNA polymerase 1 releasing the RNA primers and substituting them by DNA nucleotides, and here is the DNA ligase, okay, coming to seal it up and glue the 2 strands. And finally, proofreading. Here, DNA polymerase 3 isn't just a builder, it's actually more like a spell checker. Okay, if it adds a wrong base while working, like putting a T instead of a G, okay, it can backtrack and remove the nucleotide from the 3 end and insert the correct one. Alright, this is critical to prevent mutations. As I said earlier, any mistake in such a step is a mutation, and we're going to study mutations in simple DNAs later on. So alright, that is the full checklist for topic 1.1. We've covered the SL basics of semi-conservative replication, PCR, and we've crushed the HL details of leading and lagging. And here is just a short summary to make sure you've got everything. Pause the video, read the points, make sure you have got everything. You just recapped every information if you have already known it, and if you did not, I hope you've got at least 70%. So, and if you really want to test if you know if you've known these topics, wait for the question video related to this, and I really need your opinion on this thing. Just tell me in the comments if you would like the revision videos to be on separate topics just like this one, or on a whole theme or a whole level of organization like the whole molecules video, or the whole continuity and change video? Please tell me in the comments, and we're going to apply this to real IB questions later. And thanks for watching 45/7. Go crush your revision, and I'll see you in the next video.